You've heard about it. You've got it in your very own body. Let's know more about DNA. Well, the basics anyway. It's me, Moser again. And after this video, you should be able to identify the parts of a DNA molecule, correctly pair up the DNA bases, describe the steps in DNA replication as well as explaining when DNA gets copied, and explain what mutations are, as well as naming the 3 types of point mutations. Are you ready? Let's go. Well, first off, DNA stands for deoxyribonucleic acid. That's the D and the N and the A. And what is it? Well, it's one type of nucleic acid. It carries instructions for building cells and for performing pretty much all the functions of life. Here's an image of DNA. That's the one on the left. The other nucleic acid, which we won't talk about just yet, is RNA. We'll discuss that a little later. DNA is actually a long molecule with a very interesting structure. It starts off looking like a ladder, but it's a ladder that looks like it's been given a twist at the top and the bottom until it's a spiral. Helix is just another word for a spiral. And DNA gets called a double helix because of the 2 sides of the ladder, as it were. And when we say that DNA is a long molecule, we're not kidding. This stuff is tiny. It's like threads, but a single bacterial cell has got 1/10 of an inch of DNA in it. And one of your cells, one individual cell, has approximately 6 feet of DNA. This stuff is long. Every single piece, or we can call it a strand, of DNA is made up of the same parts. There are 2 sugar phosphate backbones, in essence. These form the sides of the ladder. And then there are 4 chemicals that we call the bases. We can also call them nucleotide, that join in pairs to form the rungs of the ladder. There are 4 bases: adenine, thymine, cytosine, and guanine. They're not really color-coded that neatly, but I've just tried to be consistent in this video. Those 4 bases only link together in a very special way. Adenine will only pair with thymine, and cytosine only pairs correctly with guanine. If you need an easy way to remember this, you can remember that cars park in garages, C and G, while apples fall from trees, A and T. The bases pair the way they do because of chemical bonds. We can think about it like a key and a lock, and only the right key will fit the right lock. Now, these chemicals are not this simple looking in reality. Actually, they look a little bit more like this, and even that's a simplification of how they look. They're a bunch of atoms joined together in a very particular way. Adenine looks like that. Thymine, something like this. While guanine looks something like that. And cytosine like that. Maybe we could just stick to basic geometric shapes. I think that'll work. So what if you want to make a new cell? We know that all living things have DNA in every cell of their body, or in their only cell if they're unicellular. But how do we make sure that a new cell gets the same DNA as the old cell? Well, there's only one way to do that. That's to copy the DNA. In fact, DNA has to be copied every time a new cell is made. For prokaryotes, bacteria and the like, and unicellular eukaryotes, that's eukaryotes that are made up of one cell, this means when they want to reproduce, they have to copy their DNA. For multicellular organisms, such as you or me, a maple tree, a mouse, an earthworm, a dandelion, any of those multicellular organisms that keep their DNA safely tucked away in their nucleus, these eukaryotes, they have to make new cells when they want to grow larger or replace cells that have been damaged or that have worn out. all of these require copying their DNA. The way that the DNA bases pair up makes copying DNA really simple. Now, copying DNA is called replication. So the first step in DNA replication is that the two halves of the DNA separate. Some people call it unzipping, but in any case, that double helix opens up. Now, a second new sugar phosphate backbone is put into place on each side. New matching base pairs are connected to the backbone and to the original bases. And when the entire strand of DNA has been copied down its length, the two strands separate. And voila, you've got two new strands, each with one of the old backbone pieces, and each with new section of sugar phosphate backbone. Wow, that's actually really simple. In truth, it's a little more complicated than that, but that'll do in terms of understanding the process of DNA replication. Now, let's talk about what can go wrong. Have you ever had to copy something? Maybe out of a textbook, maybe you were copying a friend's notes. Have you ever made a mistake? Woohoo! I have. Well, the fact is that mistakes happen, and an error in copying DNA or RNA, the other nucleic acid, is called a mutation. Now, most mistakes actually get fixed so they don't become mutations, and only mutations that are happening in reproductive cells in multicellular organisms get passed on, but it's still important to talk about mutations. An error that changes a single base or a single nucleotide is called a point mutation. Can you find the point mutation in that yellow circle? One base, just one. One base got changed. There it is. When the DNA was copied on one side, the wrong base was put in there. Oh, really? I wonder what that'll do. We'll answer that in a little bit. But first, let's talk about the three kinds of point mutation. The three kinds of point mutation are insertion, deletion, and substitution mutations. We're actually going to talk about substitutions first. In a substitution mutation, as you might expect, one base is substituted for another base during replication. And if we look carefully right there, we see that instead of a guanine being put in on the new half of the replicated DNA, the cytosine was repeated. Will that do something? Well, we'll talk about that in a minute. In an insertion mutation, as you might guess, an extra base is inserted into the DNA strand during replication. And as you can see right here, it looks like the cellular machinery got a little carried away and stuck an extra thymine into that strand. Now what's interesting is that causes a lineup problem. We'll talk more about that when we talk about protein synthesis. So that's an insertion mutation. A deletion mutation is, you can again probably guess this, when one base is left out of the DNA sequence during replication. One base gets deleted. And if you look carefully in that little yellow circle, you can see that there should be an extra guanine, but somehow it didn't get there. This kind of mutation also causes a lineup problem. Things just don't line up correctly. Well, is that a problem? We'll see. What's important to know is that some mutations cause changes to cells, some don't. That's okay. We won't really understand why that is until we start to talk about protein synthesis. But here is a cell that has a mutation that did cause a change to it. That's a sickle cell, and we'll talk more about that later too when we talk about genetics. Some mutations are harmful, some mutations are actually beneficial for the cell, and some mutations, like we said, don't actually cause any changes. Okay, you got it? Let's check. Are you able to identify all the parts of a DNA molecule? Can you correctly pair up the DNA bases? Can you describe the steps in DNA replication and explain when DNA is going to get copied? And can you explain what mutations are and name the 3 types of point mutations? If so, you're good.